Hello everyone, welcome to Anupama biology classes. This is the fifth video in the series of Human Physiology. In this part, we will discuss about the human excretory system. So let's start the topic. Here the first thing we will have to know that what is the nitrogenous waste material. And the nitrogenous waste material are the main excretory product that is formed as the end product of the protein metabolism. What is protein metabolism? Protein metabolism is responsible for the synthesis of proteins and amino acids and the breakdown of proteins by catabolism. Now, these nitrogenous waste materials are produced by two ways. First one is the deamination of excess and unwanted amino acids in the food taken, that is the exogenous source. And second is the breakdown of body's own proteins and nucleic acids, that is endogenous source. Here a word that is deamination. What is it means? Deamination means removal of an amino group from a molecule, and in human body it takes place in liver and kidneys. And the exogenous means originating from outside of an organism, while the endogenous means originating from within an organism. Now on the types of animals, nitrogenous waste excretions are of three types. First one is ammoniotalism. Ammoniotellic organisms excrete nitrogenous waste in the form of ammonia, and the examples are most aquatic animals like fishes, protozoans, crustaceans, and platyhelminths. Second is ureotelism. Ureotellic organisms excrete nitrogenous waste in the form of urea. Examples are all terrestrial species, cartilaginous fishes, few bony fishes, adult amphibians, and mammals, including humans. And the last is uricotelism. Uricotellic organisms excrete nitrogenous waste in the form of uric acid. Examples are most snakes and lizards, birds, terrestrial arthropods, including insects. Now, the various type of excretory products and the excretory organs in case of the human. You know that only nitrogenous waste are not the excretory products. Beside these, some other products are also found in our body which have to excrete. So, the excretory product and their excretory organs are here. First one is the nitrogenous waste, which excreted by kidneys and skin. Water, which excreted by kidneys, skin, lungs, and intestine. Carbon dioxide, which excreted by lungs. Salt, which excreted by kidneys, skin, and large intestine. Pigments, which excreted by kidneys and intestine. Spices, which excreted by lungs and kidneys. Drugs, which excreted by kidneys. And the vitamins and hormones, which excreted by kidney. Now the urea is 1 lakhs time less toxic than ammonia. So ammonia is the most toxic in our body. And some animals like earthworm and prawn are ammoniotelic and partly ureotelic. Allantoin, creatinine and hippuric acid are other nitrogenous waste products found in case of the humans. Now excretion. The elimination of nitrogenous product from the animal body to regulate the composition of the body fluids and tissues is called excretion. These nitrogenous waste products are formed by the breakdown of proteins and nucleic acid, and these materials must be eliminated from the body because they are toxic. So for this waste material there are some excretory organs in our body, and the organs which remove the waste product of metabolism from the animal body is called excretory organs, which are of two types. First one is the primary excretory organ and second is the accessory excretory organs. The primary excretory organs are kidneys, and the accessory or additional excretory organs are lungs, liver, intestine, and skin. Now you know that there are different types of animals are present in our animal kingdom, and they have different types of excretory organs. Like the first is in case of the protozoans, sponges, and coelentrates by diffusion through the cell membrane into the surrounding water where the solute concentration is lower. In these organisms, ammonia is the chief excretory product. Here, diffusion means the movement of particles or substances from higher to lower concentration. In case of the other invertebrates, like in case of flatworms, with the help of protonephridia, excretion occur. Protonephridia are the network of tubules lacking internal openings and have phloem cells with vibrating cilia for excretion. In roundworms have intercellular canals and canaliculi opening out at the excretory pore, and in case of annelids by metanephridia with internal openings, and these nephridia consist of nephridial canal that opens into nephrostome and to the exterior by the nephridiopore. In arthropods, like in cockroaches, by Malpighian tubules that floats freely in the hemocoel. So, what is hemocoel? Hemocoel is the cavity between the organs of arthropods and mollusks through which blood circulates. In case of crustaceans, by the pair of antennary or green glands and renal sac. In scorpions and spiders, by Malpighian tubules or coxal gland. In mollusks, one or two pair of kidney present for the excretion, and in echinoderms, gills and tube-feet present for the excretion by diffusion. Now, the excretory organ of vertebrates, and the main topic is human excretory system. Human excretory system in case of consists of first, that is the one pair of kidney, one pair of ureter, one urinary bladder and one urethra. So the first is kidneys. Here in the picture you can see the frontal section through the kidney. Kidneys are the dark red bean-shaped organs and about 120 to 170 grams in male and 120 to 135 grams in female. They are placed in the back wall of abdominal cavity just below the diaphragm and one on either side opposite the last thoracic and first lumbar vertebrae. The left kidney is little higher than the right and the left is slightly larger than the right kidney. Kidneys are retroperitoneal. It means it lie behind in the peritoneum, and each kidney has a connectivity called the hilus or hilum, which provides a place through which blood vessels, lymph vessels, nerves enter into ureters. Kidneys are metanephric in mammals. So what is metanephric? When we are developing, kidneys form in three different stages: protonephros, mesonephros and metanephros. Protonephric, which forms at 22 days of gestation, means pregnancy, and develops in the cervical region of the embryo. It can't excrete outside, hence it is non-functional. While mesonephros, where the principal excretory organ developed during early embryonic life at 4 to 8 weeks of gestation, and the last is metanephros, which are definitive kidneys and develop at 32 to 36 weeks of pregnancy, but it won't be completely matured. Maturation will take place even after birth. So it is called metanephric because it is at the last stage of development. And a kidney is composed of numerous nephrons or urinary vestibule. Nephrons are the functional and structural unit of kidney. We will describe the structure of nephron ahead. Nephrons are enveloped by capsule. You can see in the picture the arrow indicates the capsule. It is the outermost covering which contains nephrons. Kidneys have two regions: outer renal cortex in the picture and the inner renal medulla. Medulla is divided into 15 or 16 renal pyramids, each having a broad base towards the cortex called renal papilla. Each renal papilla projects into minor calyx which join to form major calyces. The major calyces open into pelvis which leads into ureter. Besides this, there are two other structures, that is the renal arteries and renal vein, which opens into the kidney. After that, ureters. The ureters are about 28 centimeter long. Ureters open into the urinary bladder. You can see in the picture, the ureters arises from the hilum of each kidney. This is the structure of kidney, and from the hilum ureter arises, which is in pair. And this ureter opens into the urinary bladder, and which is present in the front body wall. This is the urinary bladder. You can see in the picture female bladder and male bladder separately. It is situated in the pelvic region of the abdominal cavity. The muscles of the urinary bladder is called detrusor muscle. The neck of the bladder leads into urethra, and the bladder can hold 700 to 800 ml of urine. The neck of the bladder guarded by two rings of muscle fibers which are sphincters. Both the sphincters must relax to let urine pass out from the bladder. Now the urinary bladder opens into urethra. In female it is short, opens by urethral orifice or urinary aperture in the vulva in the front of the genital aperture. In male it is long, carries urine as well as spermatic fluid, and it opens at the tip of the penis by urinogenital aperture. Urethra starts from the lower part of the urinary bladder and leads to outside. Infection in urinary tract is more common in female than male because in female urethra is short. Now the structure of nephrons. It is the structural and functional unit of kidney, and on the basis of location in kidney, nephrons are of two types. In the picture you can see here the two types of nephrons. First one is the juxtamedullary nephron and second is the cortical nephron. Cortical nephron lies in the renal cortex and have very small loop in renal medulla, while the juxtamedullary nephron have the Bowman's capsule close to the junction of the cortex and medulla and have very large loop in renal medulla. You can see in the picture the juxtamedullary nephron have U-shaped structure and it is very large. Cortical nephrons are 80% of the total nephrons and the rest 20% is juxtamedullary nephrons. If we talk about the function of these two, the cortical nephrons control the plasma volume when blood supply is normal, and the juxtamedullary regulates the plasma volume when the blood is in short supply. Now the structure of nephron, which have following parts. First one is the Bowman's capsule, which contains glomerulus, and the glomerulus and Bowman's capsule combine to form Malpighian body. It facilitates the process of ultrafiltration and it lies in the renal cortex and it is the start point of the nephron. Now it opens into proximal convoluted tubule, in short we can say PCT. It lies in the renal cortex and the wall has microvilli on the free surface. In the picture you can see all the part of the nephron. First is the glomerulus, then the proximal convoluted tubule, then descending loop of Henle, then ascending loop of Henle, then distal tubule, after that the collecting duct. Now the loop of Henle, and you know the loop of Henle is a U-shaped structure and it consists of two limbs, ascending limb and descending limb. Descending limb is a continuation of PCT into the renal medulla and an ascending limb which re-enters the renal cortex and join the DCT. This is the ascending loop of Henle and this is the descending loop of Henle. Now the distal convoluted tubule, that is DCT. It lies in the renal cortex and opens into collecting tubule. It has juxtaglomerular aperture, which is a special sensitive region formed by cellular modifications which plays a role in the regulation of glomerular filtration rate. Now this opens into the collecting tubule, which are large tubes each receiving the collecting tubules of the several nephrons, and the collecting duct which runs through the medullary pyramids and open into calyces which lead into the pelvis. So this is the complete structure of the nephron. Now the physiology of excretion. Man is ureotelic, and this urea is formed in liver and a small amount in the brain and kidney. Now, the excretory process has the two main events. First one is the urea formation and second is urine formation. Urea is formed in the liver by a cyclic process called urea cycle, ornithine cycle or Krebs-Henselt cycle. You can see the picture. This is the complete urea cycle, and in this picture and the urea cycle, the urea formation consists of the first conversion of ornithine to citrulline. Here the ornithine combines with ammonia and carbon dioxide to form citrulline and water. Next is conversion of citrulline to arginine. Here the citrulline combined with more ammonia to form arginine and water, and after that conversion of arginine to urea. Here the arginine decomposes to form urea and ornithine in the presence of arginase enzyme and water, and this ornithine is set free for reuse in the urea cycle. After that the urine formation, and it has three processes. First one is the glomerular filtration, then the tubular reabsorption, and after that the tubular secretion. Now the first, in the picture you can see this is the complete structure of the urine formation, and the first one, that is the glomerular filtration. The wall of glomerular capillaries allow water and small molecules in the blood to pass through them. Then materials in the glomerular capillaries forced out by high pressure of the blood in the capillaries. So why the pressure is high here? Because the glomerular capillaries are narrower than afferent renal arteries, and this net force moving the fluid from the glomerular capillaries are called filtration pressure. The separation of small molecules and ions from the large molecules and cells in the blood is termed ultrafiltration, and this filtered out fluid is known as glomerular filtrate. The ultrafiltered contains sodium, potassium, chloride ions, glucose, amino acids along with urea, uric acid, creatinine, ketone bodies and a large amount of water. The blood is left with only corpuscles and the plasma protein. These plasma proteins are albumin and globulin. Now it enters into the tubular reabsorption, and it occurs in PCT, that means proximal convoluted tubule. It is adopted for the reabsorption of materials from the filtrate, and the cell reabsorb entire glucose, amino acids, most of the inorganic ions, that is sodium, potassium and chloride ions. Here the glucose, amino acids, sodium and potassium are absorbed by active transport, some urea by diffusion, and the rest remains in the filtrate for the removal of urine. Now this is the structure. In this structure you can see the afferent arteriole and efferent arteriole, and the 1, 2, 3, 4, these are the process. First one is the filtration from the Bowman's capsule to the PCT. After that the reabsorption in the PCT, DCT, collecting tubule and collecting duct in the and the Henle's loop also. After that the secretion, and the last is the excretion. Now the third, that is the Henle's loop or loop of Henle. You know the Henle's loop had two limbs, ascending and descending. The first wider part transfers the nearly isotonic filtrate from the PCT to the narrower region of the descending limb, which is permeable to water. So here isotonic, what is it means? Isotonic means the extracellular fluid has the same osmotic pressure as the cell. Then the first part of the ascending limb is permeable to inorganic ions and urea. The second part of the ascending limb pass inorganic ions out of the filtrate into the renal medulla, and then the DCT, collecting tubule and the collecting duct. Here in DCT the level of plasma water falls, and the posterior pituitary lobe start releasing antidiuretic hormone which increase the permeability of DCT, collecting tubule and collecting duct to water. Because of this, water reabsorbed from the filtrate and reduce the amount of concentrated urine which is produced. They actively reabsorb sodium from the filtrate by the adrenal hormone, that is the aldosterone, which makes their walls permeable to ions. Bicarbonates ions are also reabsorbed in DCT. Then it enters into the process of tubular secretion. Creatinine, hippuric acid, and foreign substances are actively secreted into the filtrate. And the potassium, hydrogen, ammonia, and bicarbonates are secreted by active transport into the filtrate. Urea enters the filtrate by diffusion. So, the removal of hydrogen and ammonia from the blood in the PCT and DCT helps to maintain the pH of the blood between 6 to 8. In the end of process, the filtrate in the collecting duct, after being modified by the reabsorption of certain substances and addition of others, is called final urine. So after this process, final urine formed. Then what is the difference between the tubular reabsorption and tubular secretion? Tubular reabsorption refers to the passage of selected materials from the ultrafiltrate into the blood, while the tubular secretion refers to the addition of selected materials from the blood to the ultrafiltrate. Tubular reabsorption reduces the volume of ultrafiltrate, while the tubular secretion increases the volume of ultrafiltrate. Tubular reabsorption occurs by the diffusion and active transport, while the tubular secretion occurs mainly by the active transport. Tubular reabsorption takes place throughout the urinary tubule, while the tubular secretion takes place in the proximal and distal convoluted tubules. Tubular reabsorption does not occur in animals that lack glomeruli, while the tubular secretions is the only mode of excretion in animals that lack glomeruli. In case of tubular reabsorption, reabsorbed materials include glucose, amino acid, ions, water, and some urea. While in case of the tubular secretion, secreted materials include creatinine, hippuric acid, potassium, hydrogen, ammonia, and foreign substances. Now, countercurrent mechanism of urine concentration. This is the structure of the countercurrent mechanism between the collecting tubule and the loop of Henle. The role of countercurrent mechanism is to excrete hypertonic urine for conserving body water. Hypertonic in case of urine means urine is more concentrated than blood. Henle's loop and capillary loop play an important role in this mechanism. Glomerular fluid in the Henle's loop and blood in the vasa recta flow in opposite directions in the two limbs of the loops, which help in concentrating the urine. Now, the first one, that is the Henle's loop. As the filtrate passes through the ascending limb of the loop of Henle, it loses to the interstitial fluid in the renal medulla sodium chloride by diffusion in its narrower region and sodium and chloride ions by active transport in its wide region of the limb. The increased concentration of the interstitial fluid drawn out water by descending limb and collecting duct. After that, the water enters into the vasa recta and is carried away. The urine leaving the collecting duct is as concentrated as the interstitial fluid deep in the medulla, and the urine is concentrated 4 times than blood plasma. After that, it enters into the vas rectum, and the countercurrent exchange in case of the vasa rectum are the endothelial cells of vasa rectum forming the walls are freely permeable to ions, water, and urea. As the blood flows in the descending capillaries of the vasa rectum towards the renal medulla, water is drawn out from the blood plasma by osmosis. Due to increase in concentration of the interstitial fluid, and sodium and chloride ions and urea enter the plasma by the diffusion, and after that, when the blood flows in ascending capillaries towards the renal cortex, the reverse occurs, that is, water re-enters the plasma and sodium and chloride and urea leave it due to a decrease in the concentration of the interstitial fluid. So, it prevents the sodium and chloride ions from the renal medulla and helps to maintain concentration gradient in the renal medulla, which assists in concentrating the urine by the loop of Henle. Hence, this mechanism operating in the loop of Henle and the vasa recta concentrate the urine by maintaining high salt concentration around the nephrons and collecting ducts, and the production of hypertonic urine conserves body water. So this is the complete mechanism of countercurrent and the concentration of urine. Now, urine. What is urine? The fluid and dissolved waste substances excreted by the kidneys constitute urine. An adult man normally passes about 1 to 1.8 liters of the urine in 24 hours. The volume of the urine depends on your fluid intake, level of physical activity, type of food taken, and the environmental temperature. Urine is transparent, yellowish fluid, and this yellow color is because of urochrome, which is found by breakdown of hemoglobin from the worn out RBCs. And the pH of urine is 5 to 8. Urine consists of water, which is 95%, and organic and inorganic substances, which is about 5%. The process of passing out urine from the urinary bladder is called urination. Now, artificial kidney. You can see the picture, and it is a machine that is used to filter the blood of a person whose kidneys are damaged. This is called hemodialyzer, and this process is called hemodialysis. In the picture you can see the hemodialysis. Here a dialyzer is present which is responsible for the purification of blood. In this, the separation of small molecules from the large molecules by the semi-permeable membrane between the solution and water, it works as the principle of dialysis. Now, the kidney transplantation. You can see the picture. Here the damaged kidneys, which are brown in color. Normal kidney have deep red color, and the transplanted kidneys are also deep red color. You can see the picture. Here, the grafting a kidney from a compatible donor to restore kidney function in a recipient suffering from kidney failure is called renal transplantation. A kidney transplant from an identical twin called isogenic graft or isograft. A renal transplant from a sibling or a close relative called allogenic graft or homograft. In case of allogenic graft, use of an immunosuppressant prevents the graft rejection by the body's immune response. So this is the end of video in which we know about the excretion, its organ, and the process of excretion. Now the next upcoming video will be on the musculoskeletal system. For more such knowledge in biology, stay tuned with this lecture series. If you understood this topic, like and share it, and for the more such lectures, subscribe to my channel Anupama Biology Classes and click on the bell icon to get notification and be the first viewer. If you have any questions, ask in the comment section and tell me how did you find this video. Thank you for the watching.